2 weeks is
Hi, everyone.
Hi, I'm fine. How are you?
I'm good. I'm good.
I opened Word to make notes during the meeting.
Yeah. Thanks for that. Sydney is currently in lockdown, so I've been at home for a couple of weeks, but I'm guessing it's gonna be.
How many cases do you have then?
Sorry.
How many cases are there then?
Like daily, people who's been getting contracted with the Delta COVID, I think it's like 100, like 150, 170 cases every day. So it's like on the rise. So basically everyone's been told to stay at home.
Yeah, I think we are, the peak here is lowering a little bit, but we're one of the worst countries in Europe now.
Right. So
I think we had 10,000 cases a day.
Oh, wow. I didn't realize it was that high as well. Oh yes, I guess for my figures, it was just from like the States, so from like New South Wales, from Sydney essentially. But I think averages for Australia, I think it's probably, I don't know, I'm gonna say anywhere from like 300 to 500. That's just that. But like mainly it's from Sydney. That's like a huge outbreak right now. So does that mean that it's also kind of like travel restriction, can't move around in where you guys are as well?
No, it's not too bad. People actually are going on holidays.
Okay.
But you need to have a negative test or a vaccination, or so you can't travel around with only your passport. You also need all these documents that you're either vaccinated or that you had the disease or that you are free from COVID at the time of travel.
Right. Hi, Kim. How's it going? She can't hear me yet. Oh,
no, I can. I'm just finishing off a quiz night that's supposed to finish at 9 o'clock, but always goes over. So I'm listening to two things at once. But I'm good. We've lost the quiz, but that's fine. We had fun.
Right.
So do we expect others to arrive as well? It's only one past, but
I'm not sure. Yeah, let's just give it a few more minutes. So is Wilson, has he finished his PhD already?
Well, he's still writing his thesis,
Okay.
but his contract's finished. Here, usually most PhD students finish their lab work during the contract, and the writing is partly done after the contract.
Okay.
So he's writing an overview, a sort of review, and the Fennery Moore paper, that's the last part he needs to add in his thesis.
Right. Hopefully we can get that going. So I've been working with Kimberly as well on the SAR with, oh my God, like the draft done. I think I would like to also have like UCL, so Mats and Dimitri to have a think about, to have a look at that, and we can have a discussion as well before I can put that into the Google Doc. But we've got like the first draft per se of that. So that's something I guess is also quite nice to have as well.
Yes, and also we, well, Ma actually checked all these compounds if they were still active, just as a precaution, so that when we start testing any compounds, we know that they are still active because they have been stored for quite a long time. But it seems that especially the EpiChem library was, well, deteriorated during storage. I know that Ben is on a holiday now, but the thing we thought was maybe it's best for them to ask to send over these few compounds we would like to test. Because that will be the most practical one to get the compounds in the assay as soon as possible, instead of first looking what happened to the compounds, why they deteriorated, because by the end, when we found out what actually happened, then we can't test anymore before we get the new ones. So we need to get the new ones anyways, but yeah, but hopefully we can ask Ben to provide, to let them send a few compounds back.
Right. Yeah, that sounds good.
Because I saw that the majority of your compounds are from EpiChem.
Yeah, so most of the one that I suggested, except for one, is actually from EpiChem. One of them is what I made, so I can like double check. But yeah, the rest of those are just all from the EpiChem libraries. So like I'm like, even if I wanted to, I don't have access to them for like just a quick check.
No, and I know Ben has, so I see Matt also joined.
Yep, I think.
Hi.
Hi, Matt. Hi,
Sorry, Mike. Were you talking about retesting samples?
Yeah, we'll just have like a quick chat and yeah, get there as well. Yeah, regarding the some of the compounds that I suggested for in vivo, but then we have that little problem with the retest giving unexpected results.
And so you have a list of compounds that you were suggesting for those, and then I guess we need to know which samples we might be nervous about in terms of degradation. Is that where we're up to?
Yes.
Difficult. Don't know. I think it's probably pretty important that we are confident of any compounds if we're talking about in vivo, right?
Yes, so that was my suggestion, to do it on the practical side, since there are not too many compounds which were suggested by the SAR, to ask Ben if EpiChem would resend us those isolates so that we can do the quick check of those few compounds in vitro and then move to the in vitro and then move to an in vivo model, just not to lose the season.
What's the sorry, I need to just remind myself of the issue number there of the compounds that we're talking about and whether we know where they're from.
Well,
Sorry,
the majority is from Epicam, actually.
Oh, so it's issue 61. Sorry, got it. Right. And okay, so the origin is Epicam, is it, for the compounds that are of interest for in vivo?
Yes. So the one that I
Except one.
Yeah, except for one of those.
Well, that simplifies it a bit, doesn't it?
I'm sorry, what was that, Matt?
I guess that simplifies it.
Yeah.
Right. Because if it's just Epicam, then it's just another shipment, I suppose.
Yeah.
What's the sorry, to play catch up here, what's the amount that we're talking about that's needed?
A few milligram, so a max of 5 milligram, but probably 2 or 3 milligram is enough.
Right. So I guess first step is to see if Epicam has the compound. Yeah.
All right, so yeah, so let's just note that down so we can get Ben to look into that further. So I've already noted that down the discussion points for the meeting as well, but let's just leave that for. So the I've got the item from previous meetings. So the ISNTD.
Oh, just on the previous thing, Ben's on holiday, so I think you need to circumvent. And I would suggest going directly to Epicam to ask if they've got stocks of those compounds.
Okay.
As a first step, if it needs clearance to go to be shipped, then we can deal with that. But just to get the conversation going, I suggest a direct email to Epicam.
Do you have direct email to them or not?
Oh, I think so, somewhere. I mean, it's Wayne doesn't work there anymore, I think, but it is Martine Keenan, isn't it? Hong, we must have been in touch with her right at the beginning.
Yes, so we've talked to her once in like 2017.
Right. Okay. If she still works, then I'd recommend email to her in the first instance.
Okay. All right. So the other item. So I'm going back to the agenda now. So the other item is the ISNTD conference. So that was for Matt to check with the organizer of the conference to confirm about the presentation of some of our research at the conference.
Yeah, where are we? That's odd. There has been a conversation. I think we already had this last time. There's been a conversation between ISNTD and DNDI, but I don't know what the outcome of that was. Okay. Nothing much, right? Have you heard anything about that, Wendy?
I didn't hear anything from it, but
okay, I'll trace that. I'll do it now while I'm listening.
Okay. So Dimitri, have you shipped that your batch of synthesized compound from that was one of the items from last time? Did that all good? So I know that Kimberly has also shipped all of the UCITS compounds successfully to Wendy as well.
Yeah, those have been shipped successfully, and they're
yeah,
already in the Netherlands.
What we did, we received all compounds, and dissolved them already, and they were added to the screening plate, and this morning the fungus was added. So hopefully the first results will come in next week. So all compounds are tested together in a high and low dosage, just to see for activity.
Okay, that sounds good. Hopefully we get those results as well. Yes, for the next thing is the SAR, which I've been working on as well. So I'm almost done with the SAR. What I would like to do is probably pass it through to have for Matt and perhaps Dimitri to have a look at that as well. I think Alice is also going to have a look at that to have some input. So Kimberly has already I've already had some discussion with Kimberly in terms of like the direction and some interesting points that we found for some of these SAR. I think what I do is just when I finish this first draft, if Matt and Dimitri can have a look at that, and you know, if there's anything that needs discussed, we can probably have another talk on that. I'm gonna probably share the Google Doc link for that as well, and then after that, I think we can then put that into the manuscript. So right now, I'm just noting down all of everything that I saw in the SAR, but it's probably a little bit too long for the manuscript itself. So I think we can refine that a bit further to just make it more concise. Is that okay with Matt and Dimitri?
Yeah, great. Are you so are you mainly doing this off GitHub and on some other document?
Yes, I haven't put it into the manuscript link on GitHub just yet, but I think I'd like to just have a second or in this case third and a fourth opinion on that as well, and then I'll just copy that over into the main manuscript.
All right, this is mainly issue 51 though, right? The thing you started on the general overview of the SAR.
Yes. So I've done basically I rearranged all of the tester compounds into group, and then I tabulate them with like the different bits and pieces, the different modification and how the activities are affected. So I've got some of those screenshots that I showed you on GitHub as well. And then basically I just did writings. I just wrote them out, what I saw. So
great. Can I just ask, I mean, the two things on the SAR diagrams that you were doing, which look very comprehensive. One was just to clarify somehow on the diagram, any compounds that, I guess I was thinking which compounds have been recently tested versus which compounds were in the original paper.
Yeah.
Is it the case that they're color coded if they, is there a certain color being used for compound which was in the original paper, which isn't new?
There is currently there's currently no color coding for that. However, I did note down that our 5 initial leads from the previous paper, I actually highlighted that. So those are like the starting point for each of the table for the current SAR right now.
Okay, all right. And then for the others, I mean, obviously most of the structures are in black. Were you using a color coding for activity?
No, I haven't actually color coded activities yet. So the apart from the all of the structures are black, I do have a general like simplify structures that is color coded with letters and color. But the actual table itself is still currently all black as a design.
Right. Right. For just, I mean, it's a cosmetic thing, I think. I'm just looking at the structures now. For the paper, you just got to make sure that your drawing style is consistent. You know, sometimes you've got the benzene ring with a circle in it, and sometimes it's with the double bonds.
Yeah.
Just make sure it's consistent. I would advise the double bonds. And also, I think you've got this done, which is that all the compounds are drawn in the same orientation. So I think that's looking fine.
Yeah. Okay. So I guess the issue with that is I just basically copied the smart strings over.
Right.
I didn't actually change all of the benzene ring to the one thing. But is it required to have the actual structure in the paper as well? Because I was thinking we can, because I tabulated the data, which does have all of the structure in it as well. Is that sufficient for the manuscript?
You should put the structures into the SI anyway. Okay. I would suggest anyway, either where you have the experimental procedure or somewhere, just so that they're drawn and people can go and look at it and there's no confusion. So you would probably have to do those things anyway. And then if we use them in the paper, we can.
Okay.
Yeah, I agree. I mean, it's nice to have them all drawn out like that, even if the paper manuscript itself would only have a subset.
Yeah, and it'll be good for GitHub to have those things and it'd be clear as well, I think.
Yeah.
Great. Okay, great. No, I mean this analysis is a little overdue, actually. I mean, as a group, we've got to keep a good eye on, what is giving us activity and what is not. The thing that struck me actually was how many compounds there are there. I didn't actually realize we tested so many. In fact, it's kind of crept up on me.
Yeah, I think from what I did, there was I counted 148 like unique structures. That means that there's also like one structure with multiple testers as well. So that was 148 unique structures that we tested.
You mean tested by Wendy since the first paper?
Yes.
Wow. Okay.
So it's basically everything that's on the master list at the moment. It's been tested.
Yeah.
Well, with exception to the new ones that we just sent the latest batch.
Yeah. So with the new things that we've also sent as well, that's more than 115, probably like 170, 18-ish.
Yeah, that's also the reason why we can't test them all in vivo. That's simply too much work. In vitro, that's not a problem, but in vivo, it's too labor intensive.
Yeah.
Great.
Yes. So the other things for the manuscript, which is the the chemical synthesis and characterization. So I will have to do that after I finish with the SAR. However, there are some a few characterization data that I would like to include in the manuscript, but right now Sydney is in lockdown, so that's going to be causing a little bit of a delay. I'll put in as much of the data that I have already as possible, but yeah, that's just what it is. I'm currently can't access campus, so that's going to be a little bit of a problem. I've already sent the data, but to the facility for testing, but I haven't actually got anything back yet. I will chase that with the people, but that's just, heads up.
So just a note on that, and Alice has mentioned it in the chat, that there's a lot of talk at the moment that they're going to announce an extra 4 weeks on our lockdown. And the University of Sydney has got a new VC this week as well, and he's taking this very seriously. So they've really reduced the number of people who are actually allowed to be on campus. And fair enough, because it's the Delta strain and it is starting to get out of control. So I think it's going to be quite a while before we're able to go into campus and access anything. So we apologize for that, but we hope you understand.
Yeah, I can completely empathize, having been through the same thing.
Yeah.
You mean like even for postgraduates as well?
Yeah, so we are looking at getting approval for some people to be able to go in, but that's going to be some like technical staff, and they're limiting it. So there's still discussions on who's going to be allowed to go and who is not going to. So some students, like honors students and that, who are supposed to be finishing at the end of the year, might get to go in and things like that, but they haven't worked that out yet. So
we were allowed to use the labs if we were doing anything related to coronaviruses. And so
maybe that will be
started struggling to think of research projects that were virus related.
So they could go in, yeah.
Priority.
Yeah.
Venarials and COVID.
Mm-hmm.
So that's all. That's most of the points that I've already jotted down for the meeting. So I guess one thing is I put in a column in the master list just to as a note for a retest. So I've got a retest column. That was one of the comment or the discussion that we had from last time. There's only like 2 compounds in there that currently has like a retest noted on the column, but I think that might work for our purpose of distinguishing those as well. And if you have time, you can have a look at that, and if there's any problem, you can comment or give suggestion as well. But I hope that fix our problem with the sometimes compounds have the same codes and everything, but it's just a retest.
Okay.
Yeah. So apart from that, is there any other things that we need to do in terms, especially the manuscript as well, or any other things that we that will need our attention?
So I have some news to share, but it's on series 2. So if anyone else has anything else to share on the manuscript or series 1 first, I'll let you guys go for that.
So we are testing now currently the first in vitro susceptibility. So we will add that as soon as we have data. For now, I don't think we need anything else added in the table. When we see activities, we will try to proceed as quick as possible to the larvae. But yeah, we will have to wait and see first which ones will have activity and which ones not.
That sounds good, Wendy. I'm excited to find out about how they go and share that with the students who made them.
Yeah, the students will be really excited for that as well.
Yeah. So the other news that Alice and I both had a chat with John from IMAS, the Maths and Science School, who has been a part of open source mycetoma before. And he's interested in being involved again and doing something related to what we had done before with Daraprim and possibly some open science stuff. So we had a chat with him and I shared with him the work that we've done on the series 2 with the first year students at Sydney. And he's taken that away and he's going to have a look at that and see if he thinks he'll be able to do some of that with his students. So we haven't heard back from him yet, but hopefully we will soon. And he might be keen to get involved on some of the series 2. And if we have some of the data back from Wendy from the testing, we might be able to explore that a little bit more in terms of the SAR for that. But yeah, just to keep you guys updated on that little bit.
Sounds great. Good that there's so many students willing to participate as well.
Sorry, I missed this, Kimberly. How many of the series 2 compounds did you end up making?
I think
you said previously 10, I think.
I think we made 12 actually, because I think the students made 10 and then I remade the HIT compound and the demonstrators made a compound as well of their own as part of their training sessions for running the labs. So I think we sent 12 altogether. And we have materials to make 3 other ones as well, which I was working on, but yeah, I can't go in the lab. So, but with John,
the chloro and the fluoro and the methoxy scans. That's what you're doing, right?
Yeah, and we have the methyl starting materials as well to look at that at all 3 positions. Right. But for John, we were also thinking that we might look at the ring system at the other side. So maybe we can play with that a little bit. So removing the methyl group and putting some halogens and things on there and see how that goes. But I'm not sure how many he'll want to make or anything yet. So we'll see how that goes.
That's great. Do you know how many of those molecules are new to, the world, to science?
Yeah, so only the HIT compound that we originally found, that's the only one I found on PubChem.
Right, that's awesome.
And SciFinder as well. So I believe the rest of them are novel. Right. So yeah, and we got high-res mass spec and everything for those. So we're pretty confident. I think I'll do some 2D NMR as well. I did 2D NMR on one of the structures just to make sure I was pretty confident on that, and the other ones are all very similar. So I'm happy with that.
Yeah, great.
So yeah.
Wonderful.
Alice has just mentioned as well that the collaboration we're doing with the University of Washington in Tacoma, they were keen to do some synthesis too, but with their restrictions and everything, that hasn't come to any fruition yet. So that's another thing to keep in mind. We might get them working on some of the series too, because that seems to work quite nicely for undergrads.
Does anyone know any news from University of Washington regarding the Series 1 phenomenals that are on the master list? There are quite a lot of that they have picked up like to make previously, and it's been like pending for quite a long time?
I think we might have to ask Ben, because I'm not sure who the contact is with that, because we have Meg and I think Laura is the other lady's name, but I'm not sure who's previously been communicating. So maybe we check with Ben who that is, and then we can follow up with that. Oh yes, Alice has just suggested that we could ask Meg and Laura while Ben's on holidays, and then we can see who that was and try and follow that up for you. Because they do have quite a few, don't they?
Yeah, they have quite a lot actually in the master list.
Kimberly, just going back to the series 2 compounds again, were any of these fluorescent in any way? Do you know? Do they have unusual properties?
We didn't really look at anything, and all the colored, like everything was colored, but that all got washed away when we precipitated out the compounds.
So you didn't see any sort of weird behavior on TLC or anything?
Not really for any of them, no.
Right.
I can go back and check the student lab books, because I think all of them have inserted photos. They're not great, to be honest. Like columning them wasn't fun. They're just not very soluble. They're really soluble in DMSO, which is great for our testing, but they were not soluble in anything else. Like that was almost the only thing I could get them to dissolve in, sort of thing.
Right. Right.
So yeah, they weren't great.
Some of the ortho substitute analogs might be more soluble.
Yeah,
because they're a bit more twisted.
We did see differences in the solubility too. I think the para ones were the worst. I think that was especially the para ortho.
Right.
The para methoxy. That was the one I had the most trouble with.
So I think still interested. I think Wendy's certainly interested in molecules that have a fluorescence property. From the point of view of perhaps tracking where the molecules go. In terms of the grain structure. So if we had an active molecule that was fluorescent, it would be pretty interesting, because then we could track it more easily. So if that's ever seen, then that's particularly useful.
Yeah, I'll keep that in mind.
All right. Is there any other points that anyone else want to discuss for this meeting?
Just to mention that I think Mandrake renamed some of the repos, right? So now we have series 1 through 5.
Yep.
To try to keep on top of things. I think just to order things. That's not to say there's much in series 3, for example, but it's good that we've done that, so that if another group of molecules comes along, we know it's going to be series 6. Just housekeeping, basically.
Okay, that sounds great.
I had a little play around with series 5, just because we'd found some good hits in that. So basically I went through and I double checked, but I used the ones that Wilson came up with. That was all of the benzolimidazoles from the different libraries we've tested, except I think for the one that we didn't have all of the structures for. And I just did some quick analysis. There's not a lot of compounds anyway, but just to look at some of the characteristics to see how they aligned, and there was no relationships between any of them. But the carbamates look really good. Nelly, we've tested 4 of them, and 3 of them were below that 20%. So what we're considering complete inhibition. And I think that's somewhere for us to start if we want to start exploring another series. That might be very promising. Just as a note that, like, I've been looking at it a little bit, but I won't get to do anything on it. But it looks like it would be a good place for us to sort of move to next if we decide we're finished with series 1, or if we have new students who jump on board, or someone else who wants to come in.
Yeah, I agree. Those compounds belonging to that series, they all had good activities. And even though some of them were not selected for further screening, there were also a few of them which did prolong larval survival at day 4, although not at day 10. So it seems, yeah, there seems some potency in these compounds.
Yeah.
And at least they worked against more than one causative agent. That's also a good thing, I guess.
Yeah, I think we saw that all the benzoimidazole carbamates had really good in vitro activity to like where we can't distinguish between them because they're all sort of the same. So we will have to go to in vivo to look at that and sort of see, as Wendy's talked about, how they prolong survival and for how long and things like that. But I just haven't looked into that data yet. I've only looked at in vitro.
I mean, the first question about those two compounds that you highlighted are where, you know, what's the context? Where do they come from? And are there more of them? That would be the, you know, first. So they've got MMV numbers because they were in the pandemic box, I think you said. And so you think, well, okay, there are two there, but are there more that we could get our hands on that exist already? You know, that's always the first question, isn't it? Does anybody know that? You know, why those compounds are there and if they're part of a bigger set?
I don't know, but
well, fenbendazole. Oh yeah, fenbendazole was used in animals, I guess, and a few of them were. I don't know why they were selected, but they had some drug properties, and capropamide is actually one of the compounds was an antifungal compound from agriculture.
Yeah, I guess I was thinking about the two compounds that I put in the chat window, the link there that she comments that she found with the imines there. I mean, I don't know if they seem to have activity. I just haven't seen that structure before. I just wondered what those are, where they're from.
yeah, I'll look it up and try and follow through and see if I can find out where they're from. Because if they have MMV numbers, they should be online somewhere, right?
They should, yeah. And you know, the compounds in these boxes come from, well, contributors of some kind. I mean, they're not necessarily public domain, but there's a chance that they are part of a published or known set. So we might be able to get our hands on more of them.
Yeah, we also asked Curran of MMV, and she could give a few more information, but not for all compounds. Some were restricted, others were free to share.
Yeah.
So I will check what she provided and see if I can find out a little bit more as well.
Yeah, I mean, the benzimidazoles themselves, there'll be loads of those. I don't know where we get them from, but of course it's a much more explored motif. So yeah, I mean, exploring around the fenbendazole core, I'm sure there'll be lots if we find the right person.
Right. That sounds really great. Is there any other things? Anyone else? All right, so I'm just gonna draft an email for Martin from Epicame now. So like after the meeting, is that basically we're just requesting more of those suggested compounds? Is that right? We just want some more?
Yeah, I think that's the quickest and safest option to, if we want to analyze those in the larvae as well, that we get some fresh compounds for us to test if they still are active in vitro, and then if they are, which I assume they are, because I think it's due to the long storage time, because we store in plastic and you of course store in glass, so that might be different. So if they are retested and still have the activity we expect them to have, then we can test them in this larvae season to finalize the story on the Fennery moles. Because it would be weird, I guess, to have SAR in it without having done anything with the compounds identified by it.
Yeah. All right. I'll send those IDs of those compounds over to Martine and see if she's still has some of that.
While the testing is ongoing, are we putting the series 1 phenomenals on hold, like in terms of synthesizing new molecules? What should be like the future strategy for this?
I think what would be really nice is for you and for Matt, so I'm gonna send the link over. But have a look at the SAR. I think it does give some more clue as to whether we should do some more investigation in terms of the modification, because especially for one of the subset of pheneromals, I think, which is one of the compound that Dimitri you resynthesized. That one was actually a new lead that's sort of like look different from the other ones. So I think it's sort of like an area that we can focus more on, or focus or have a little bit more of an exploration in there as well. I think it would be quite useful for all of the chemists to actually just have a look at the SAR and see what we can draw out, because I might miss something. I can't see everything, but hopefully.
Yeah, we'll double check this again. Yeah. Sure. Yeah.
Just from my, me remembering what we looked at. So basically the 2 rings seem really strong. And then that third one that we've been making heaps of modifications to, having the piperazine there seems to be the most promising thing, and then making modifications off that ring. So I think if you have anything that's sort of still got that similar structure, then go ahead. But yeah, I think you have to have a look at the rest of the stuff and see. I just don't think there's much guidance for what we should start looking at. So we would still just be playing around in the dark. And given we've done 150 odd compounds, just when do we call that? And I really think that the series 5's looks quite good, and maybe it's time we start putting most of our manpower into something else. It's hard to call quits on something. I understand, especially when you've worked on it for most of your PhD and that. It is hard, but we just need to think about using our resources to the best possible.
There is actually a reason why I asked this, is because I found more molecules than have been sent recently to Wendy.
Yep.
So kind of have already them some suggestions on the table. Like the piperazine extension again, but I looked at all the actives, so all the decisions that have been made, or at least put on the table, are based on the actives that we identified previously.
Yeah.
For example, heterocyclic ring extensions and so on. Yeah, but we'll have a look at this again, so probably when I'm back.
Yeah, feel free to chuck your suggestions up, and we can all have a look at them as well, and put our 2 bobs worth in if you want it.
There is at least one new molecule that has been included in my latest screen, in the latest screen, which is an extension of the active piperazine. So basically it has the same structure, but the nitrile functional group attached. Yeah. So this will be tested basically now.
In the batch now. Yeah.
Yeah. Yeah.
Yeah.
Yeah, I think with that as well, as I think apart from the piperazine, the tri-arrow is also quite promising as well. We just haven't looked at it as extensively as we did for the piperazine and the piperidine motif. But most of our highly active in vitro, I think I put an Excel table. I think it's about 80% of all of the tri-arrow that were tested was actually quite high activities as well. So that if we do want to have a less stuff, let's look at that will also be an avenue for us to look at as well.
When we're doing some of the summaries of the SAR, which I agree is the next step here, to try and deduce what's good and what isn't before we plan too much more. The wiki for pheneromals is quite out of date in terms of the biological evaluation, and the biological data not yet incorporated into the wiki. So if anyone's making diagrams or wants to collate everything, where data are and what data needs folding into the wiki, and links to pages where analysis is happening, please do try and install those on the wiki so that we try and have one place which is up to date.
Yeah.
just related to that, I guess, we have not yet gotten any data on metabolic clearance of these compounds, do we? I guess we've never really gone that direction. There must be data in the Epichem sheet already for many of these compounds. I was just wondering about the solubility issue. We're measuring log D values, and that's because we're kind of assuming that log D helps in terms of the in vivo activity. But the other reason for measuring log D is because that can sometimes help if you have highly soluble compounds to make sure that they're not cleared quickly. In terms of these compounds, I can't remember if we're talking about, Wendy, are we talking about topical treatments or are we talking about orally available treatments? What is the sort of desired end game for a medicine?
So the desired end game would be an oral drug, simply because it's not, it's a subcutaneous disease which can enter all the way into the bone. So with the topical treatment alone, I don't think you will be able to reach far enough. Currently, the people are also treated with an oral treatment because they have been treated usually for more than 2 years. An IV treatment would also not be a very good choice. IV should only be done if we can get a very good super drug where we can clear the whole infection in a week time. But I guess that will not be realistic for now. So we should aim for oral.
But that does mean that metabolic clearance is relevant then. So we should try and just figure out if we have data on some of these compounds already from the Epichem sheet, which I think we do. Because they were evaluated for something else.
Yeah.
So we might want to dig up some numbers if they're there, and then see whether or not there's a correlation between if you have a soluble compound, a more soluble compound, if the metabolic clearance data are better, which can sometimes happen. It might add an extra reason for lowering the log D if that's the case.
Yeah, so I'm not sure like particularly about all of our compounds that we've tested right now, like their metabolic clearance, but
no, we don't know that.
For the papers that Martin has done on Chagas and Phenaramos, the 2013 and 2014 papers, they've done a lot. They've got a lot of pharmacokinetics data and cytotoxicity data on those as well, which are part of the Epichem library. So I think there is a high likelihood that we can also have those that might be able to access those data as well.
Yeah. So if there's a plot that could be done of Clint or half-life versus log D to see whether or not that's helping, that would be very useful.
Yeah.
Because it just means that low log D is doubly useful then. If anyone wanted to try and dig that up, it's an important thing, then we mustn't lose sight of that as being a criterion, I suppose. Yeah.
Okay. Okay, so is there any other business that anyone want to discuss?
Next meeting, I will not be joining because I will be on a holiday then.
All right. I hope you have a great holiday.
Sorry, when is the next one?
I just hope I will be able to be on a holiday, but I will be off anyway, so.
Yeah, okay.
When's the next one? Sorry, is it 24th? When's the next one?
Yes, it's
The fourth
I will
Tuesday.
be away.
Fourth Tuesday. So it's
Yep.
24th. Yeah, also not here, I'm afraid. Advance apologies.
Okay, no problems. Okay, so if there's nothing else, then I guess we can wrap up to this month's meeting. Thank you everyone for coming. I will send that email to my team like now, so we can get that hopefully reshipped, resent, so we can get on our way with more testing as well. Hope everyone a great day / night / lockdown, and I'll see everyone soon. Next time is going to be UCL hosting, I think. No, it's going to be someone from Erasmus and UCL for hosting and minute taking. So yeah, just we'll put just double check the roster, but yeah, I think that's what the arrangement is going to be.
Got it.
All right.
Bye, everybody.
See ya.
Bye.
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 See ya.